Okay, so if you remember last week, I don't need my today. I don't need today. I don't need this. I don't need that. I don't need word. I don't need you. Oh boy. I think we are missing 1%. But if you remember,
Kaisen Chinese audition is coming.
okay. Oh, then let's start. This thing. They don't provide any explanation in the book of how they can give in the information that the concentration of A and B is 10 to the power of minus 9 molar, and if the equilibrium constant K for A plus B associated into AB is 10 to the power of 10 liters per mole, then at equilibrium there will be 270 of A, of B, and 730 of AB complexes. Have you thought? We are not going to calculate everything, but have you thought of a way you could calculate? There are hints in the book that will enable you to calculate this. Have you thought of a way? Have you thought of a way to do that? No. No. Okay. So how, tell me, from what you have learned before, what is the relationship? So what we have is K. We have K, right? The equilibrium constant is 10 to the power of 10. The concentration of A and the concentration of B is 10 to the power of minus 9 molar. Okay? And this is in liters per. So, what have we seen before in which these are associated into the same equation? How can you put these into the same equation? We have seen something like this. How do you calculate the equilibrium constant? How do you calculate the equilibrium constant? Especially for this reaction, right? The concentration of A plus B gives AB. Okay. How do you calculate the equilibrium constant?
Concentration of AB divided by concentration of A plus B.
Very good, yes. So the concentration of the complex divided by the concentration of A multiplied by the concentration of B equals K, equals 10 to the power of 10. Okay? So, you already know that. And you already know that. What you want to know is the big unknown here. The thing that you don't know is this one. So the rest should be relatively easy. But there is a trick. There is a trick. I don't know if you have there is a trick. So can I write concentration of A, concentration of B, it's 10 to the power of minus 9. So this should be the concentration of AB, what you are looking for, divided by 10 to the power of 9 times, this is the concentration, right? This is the concentration, so this is the concentration, times 10 to the power of minus 9, same, equals 10 to the power of 10. And the concentration of AB becomes X, what you are looking for in the equation. Is this correct? This is the main trick. But if you calculate it like this, will you get the answer or not? You have to think very hardly. You have to think very hardly. Is this correct or not? Think very hard. Especially read what you have in the book. You have to be very careful. Consider 1000 molecules of A and 1000 molecules of B in the cytosol of a eukaryotic cell. This you don't care for now. The concentration of both, so the concentration of A and the concentration of B, will be about 10 to the power of minus 9 mole. If the equilibrium constant K for A plus B gives AB complex is 10 to the power of 10 liters per mole, then at equilibrium there will be 730. Is this correct or not? What I want you to tell me is, can I just use this? Using this, replacing the concentration of A by this one, the concentration of B by this one, and calculate the concentration of AB. This looks very simple. But is it really correct? If it is correct, that's okay. If it is not correct, why?
Isn't correct.
Why?
Because some of A and B is transformed
Into the complex.
into
Yes.
the complex.
Exactly. Because what they tell us is at equilibrium, this is at equilibrium, right? At equilibrium. So the concentration that they give us is not at equilibrium. It's the concentration before the reaction. Okay? So we need, and this is the main trick, the rest is only mathematics. It's easier sometimes to make a table. Okay? So there is the reactant, and the product. And then you have A, B, AB. Okay, this is for the equation A plus B, AB. Okay, I have written everything here. So, the products and the reactants. So how many of the reactants we have? What can we feel? So this is the concentration of A, concentration of B, concentration of AB. This is before the reaction, this is after the reaction. So this is before equilibrium, this is after equilibrium. So before equilibrium, we have 10 to the power of minus 9. Before equilibrium, we have 10 to the power of minus 9 more. And the reaction has not started yet, so this is nothing. But the products at equilibrium, we don't know. This is what we are trying to find out. So what we are trying to find out is at equilibrium, what will be the concentration of AB? This is what we are trying to find out. So this is X. This is the unknown. Therefore, as you said very rightly, if there is an amount of AB at equilibrium that has been formed, then there should be less A and less B. So the concentration of A and B should be what? At equilibrium, when you have X, AB.
The concentration of A is 10
Is?
the concentration of reactants minus X.
Yes, exactly, absolutely. 10 to the power of minus 9 minus X. B, what is the concentration of B?
The concentration of B is the same.
It's the same, exactly, it's the same. 10 to the power of minus 9 minus X. So then, how should you write the equation of the equilibrium constant that I gave you before? So you have the concentration of AB, but let's just write X divided by 10 to the power of minus 9 minus X times 10 to the power of minus 9 minus X. And this is equivalent to 10 to the power of. And what you are looking for is X. So this is the right start of the equation. Okay? So, let's proceed a little bit further. I have my little copy. So, what's the next step? What's the next step? What's the next step of such a monster? What's the next step of such a monster? Well, this is key, right? This is key. What's the next step of such a monster?
Just solve. Just solve.
Just solve. Yes, just solve. So you end up with a quadratic equations where you have x squared plus nantoka times x plus, this is a simple quadratic equation, and you just need to solve X. So the rest is just mathematics. But the key, and that was very, very well done, the key is this. What we are given is the concentration of A and B before the reaction. So at equilibrium, the amount of complex remove some A and B. So we need to calculate the concentration of A, B here based on this. So that was the trick of the whole thing. All right. For today, that's enough. Enough mathematics. Now we are going to finish the chapter 3 and start the chapter 4. If you want to see later on, I have written all the things if you're interested. Okay, so the last bit that we saw last week was these activated carriers that catabolic reactions provide energy, and the energy is captured in activated carriers that is then carrying the energy where it is needed, but also when it is needed, which is the advantage of having these carriers. And then we are going to see briefly some of these carriers, and of course the one that is the most common you already know. This is ATP. As I explained before, ATP has two high energy bonds. They are called phosphoanhydride bond, here and there. And it can lose these bonds, and the delta G is negative, so the energy is available for another reaction. But then it can be recycled into ATP, and this needs energy. Okay, this is why we eat, because we need to regenerate our ATP so that it can be used for other reactions. So this is the main thing. But ATP does not only provide energy. Sometimes it provides a phosphate. Okay? So you have ATP, and the last phosphate can be added onto another molecule. For example, here we have a hydroxyl here that is then phosphorylated with a phosphoester bond by, for example, protein that will use ATP to add the phosphate onto this molecule. Typically, proteins are modified like this. Okay? So ATP is not only used for energy, it's also used as a carrier of phosphate to add phosphate to target molecules. And energy stored in ATP is often harnessed to join two molecules together. Look, for example, at this equation, which is AH plus BOH gives A plus B plus H2O. Alone, it will not happen. But thanks to ATP, this reaction will be favorable because ATP, in a very nice way, is first added, a phosphate from ATP is first added to the molecule B here, and this molecule is activated. We say in English it is activated. It's a high energy intermediate that can be used in the next part of the reaction, in the real condensation step, where you have A, and the O and phosphate here will actually be changed to make a bond between A and B. And this, directly, it's not possible to make a bond, but thanks to this high energy intermediate with the phosphate coming from ATP, this reaction is possible. This is an example here where you have glutamic acid, which is an amino acid with this side chain, okay? And then you have an activation step using ATP, which gives a high energy intermediate of glutamic acid with a phosphate here. And then in the second step, which is the condensation step, then there is an amine that is then added onto the same carbon here. thanks to this high energy intermediate synthesized with ATP. So not only it provides energy, but it really adds the phosphate onto a molecule to make it a high energy intermediate. Then, okay, that's energy and phosphate. But if you think catabolic and anabolic reaction, this is oxidation and reduction reaction. Okay? Catabolic, you are oxidizing your substrate. Anabolic, you are building something. So you need to reduce. So in the cell, there are carriers of exactly this type. Activated carriers of electrons, activated carriers of electrons. And the two main one is NADP, NADP plus, NADPH, and also NAD, so NADH and NADPH. So when you add H, it means it's reduced. Okay? So these two couples, they are very similar. So you can see what is written here. This is the structure of NADP plus. And you see it's a nucleotide. It has ribols, it has phosphoester bond here, and it has two bases here. And if there is a phosphate here, this is NADP plus. If there is no phosphate, it's NAD plus. Okay? They have very, very similar structure. And what you can see is that they are both used for the transfer of proton and electron. So for example, NADP plus is lacking electron and proton. So it's actually used to take proton and electron from a donor molecule that will become NADPH plus H plus, and then this can then be given back to another molecule. So it's a couple. It can be reduced and it can be also oxidized in an ending cycle like this. Okay? The problem is, well, there is no problem, but why are we having NADP plus? This is NADPH, where you can see that actually what you get is not only one electron, what you get is a hydride ion. It's a hydrogen atom plus one electron. And then you can see the structure of this ring, the nicotinamide ring, is actually changing. It's very good acceptor and donor of electron because of all these double bonds that can stabilize even when there is a plus charge here. So why are we having two of them? Why are we having NADP plus NADPH and NAD plus NADH? It's because they are used for completely different purposes. And NADP plus, it's written in the book if you want. It's on page, if it's easier to follow. It's on page 110. It's on page 110. NADPH is used mainly in anabolic reaction. Is used in anabolic reaction, while NADH is used in catabolic reaction. NADPH, anabolic, NADH, catabolic. So because of that, what do you think is the main form of NADH? And what is the main form of NADPH? Concerning what I told you just now. NADPH is used in anabolic reaction. NADH is used in catabolic reaction. So you know there is a couple, okay? There is the oxidized form and there is the reduced form of both of them. If NADPH is used mostly in anabolic reaction, what will be the major form of NADPH? Would it be NADP plus or NADPH? If it's used in anabolic reaction.
NADPH.
Why?
NADPH should reduce other elements.
Excellent. Exactly. So NADPH is used in reducing reaction, in anabolic reaction, which are mainly reducing reaction. So it has to be kept. The ratio NADPH, NADP plus is very high. So that there is a lot of electron and proton that can be given to molecule to reduce them. NADH is the opposite. NADH is used mainly in oxidizing reaction. So the main species of NAD is NAD plus because it's actually an oxidizing agent. It will oxidize molecules. So this is why we have two of them. One is a reducer, one is an oxidizer. And this very specific phosphate here, although the chemistry of the molecule is pretty much the same, having one phosphate here or not will actually change the shape a little bit of the molecule. So some enzymes will associate only with NADPH, some enzymes will associate only with NADH, just because of the different phosphate here. Okay? So they will use by different protein, by different enzymes to mediate different reaction, and it's mainly thanks to these having a phosphate there. For example, one of the reactions in which NADPH is used as a reducing agent is when you have 7-dehydrocholesterol, and you can see here there is a double bond between these two carbons of the cycle, but these carbons are being reduced by NADPH, and it becomes cholesterol. This is one of the main reactions of NADPH mediated reduction of a molecule. Okay, so this is what I told you already. NADPH is used in anabolic reaction, supplying energy-rich electron. NADH, special role in catabolic reactions, producing ATP by oxidation of food. So the ratio NAD plus NADH is kept high, the ratio NADP plus NADPH is kept low. So this is the same thing, saying the other way. But there are many other activated carriers that we are going to see here. ATP, you already know, it's a carrier of phosphate and of energy. NADH, NADPH, and there is another one here, FADH2. These are donors or acceptors of electrons and hydrogens. You may ask the question, we have another one here, FADH2. And actually, FADH, it's a little bit like NADH, but it's more powerful. So if there are reactions that need more oxidizing power, then FADH will be used instead of NADH. But we also have a molecule called acetyl coenzyme A, which gives acetyl group, which we will see later what it is. But there are other carriers, like biotin. It's a donor of a carboxyl group. We have S-adenosyl methionine, which is a giver, it's a carrier of methyl group. And we have uridine diphosphate glucose, it's a carrier of glucose. Now, this is very funny because we have ATP, we have NADH, which we saw was a nucleotide, we have adenosyl methionine, it's again a nucleotide, and we have uridine, it's again a nucleotide. Why it is that many of the carriers are nucleotides? Why do you think that is so? What is the reason why in our body, most of the carriers carrying various molecules from methyl to phosphate, these are nucleotides? What kind of thing would you think about when you know that? From what we said previously about the origin of life. Do you remember about the molecular origin of life? What came first, DNA, protein, or RNA? the theory. We don't know for sure, but what is the theory? Life started as what? RNA? Started as protein or as DNA?
In old age, no, there are no protein, there are RNA.
Yes. So that's the current theory. The current theory is that the very, very beginning of life was like some sort of RNA molecules that could, in the same time, catalyze their own copying. Because RNA is a nucleic acid, so it can still do base pairing, but it also has enzymatic activities like protein. So what scientists think is that the fact that many of the carriers are nucleotides, it's also a kind of indication that they have been there from the beginning of life. Since the chemistry of life was based very early on RNA, on nucleotide, not deoxyribonucleotide, this is a sign that really life may have originated as RNA-based chemistry. And so this is what, well, it's not a proof, it's just an indication that somehow the chemistry of nucleotides is very important. And these groups, they are conserved everywhere, from bacteria to human. We all have these things. And this one, it's an interesting molecule, which is here, right? The donor of acetyl group. You see, again, it's a nucleotide, okay? And adenine, ribose, 2-phosphate, and then a chain of carbon, blah, 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 blah. And it ends up with a very small group that is called an acetyl. And of course, there is a high energy bond here that can be given to other molecules. And it's very, very funny that in all cases, what is given is a very small part of the molecule. It's kind of a big molecule, not so big, but compared to that, this is very big. Before as well, right? This NADPH, NADH, it's kind of a big molecule and only a very small part of it is transferred. So why do you think that is so? Why do you think that these carriers are actually fairly big for the small group they are carrying? What could be the reason for that? What could be the reason for these carriers having quite a substantial part that is not really needed for the reaction here? We have a high energy bond, maybe that depends on the sulfur here certainly, because the sulfur is quite electronegative, so it will pull up the electron of carbon, so this link is easily cut. And maybe this is kind of important for the chemistry of that, but it's too far away. So what is the advantage of having such a big?
If they are small, they when they release high energy part, then they become completely different shape or structure.
Yeah, that's right. So they have such a big structure because when they lose this acetyl, especially this one, when it loses its acetyl group, but it remains coenzyme A. Okay, this is a relatively stable structure that can be regenerated, and the new acetyl can be added on the coenzyme A. Something else, another reason is also possible. That is kind of linked to what you said. Yeah, as you said, it cannot be just sulfur and a hydrogen, because then if you cut this bond, you just have SH. It will not, it will just react with many things. So you need to have a solid base. But what else? What could be another reason?
For enzyme A is used for finding a specific molecule.
That is interesting. That is interesting. It's using, yes. So I think what you want to say is that this is actually a handle. These enzymes will bind to these things and use these carrier to use what they are carrying. So this is just like a handle where some molecules can take these guys and react with them. This is they react, they will bind to many type of protein and then they will be able to react. So the image is like a handle, like the handle of your bag. It's something that proteins will, this is what a handle means, you know, like a handle. Handle. Okay, so you have maybe an enzyme here, this is a protein, and then you have the coenzyme A here, that is inside here, something like this, and then this part will be the part that can be bound by many other molecules to react with. And the very little tail here is used for the reaction, but this is very important for recognizing what it is. It's a kind of handle for that. Okay. That's another one. That's the one here. This is the biotin. Biotin. Have you heard of biotin before? Have you heard of biotin? Sometime when you go to the hospital or when you go to the physician, they may talk about biotin. No? Biotin is a vitamin. It's something that you must eat because you cannot synthesize it yourself. And what is biotin? It's a small molecule like this that is attached to some enzymes. For example, this pyruvate carboxylase enzyme. And then this biotin is used as a carrier of carboxyl group. This is carboxyl group, right, which you know, C double link O and then O there. And in this case, the carboxyl becomes from bicarbonate, which is not organic molecule at all. So you have bicarbonate that is used in a reaction to bind to biotin, and then you have a carboxylated biotin here, and then this enzyme with biotin that is carboxylated is then used as a carrier of this carboxyl. And for example, in a reaction in which pyruvate is transformed into oxaloacetate, you can see that the pyruvate gets carboxylated by the carboxyl carried by biotin. Oxaloacetate, maybe it's very, you haven't heard before, but it's a very important molecule in the metabolism of sugar in our liver, where we use pyruvate and we synthesize oxaloacetate to synthesize sugars. So these are pretty important reactions, and they all use carriers. But if you remember what we saw before, that nucleotide gets added to each other to make DNA, to make RNA, amino acid, they get attached together to form proteins, and so on and so forth. And this requires energy. Okay? This requires energy. You have a condensation reaction. For example, you can think about whatever it is. It can be two nucleotides, it can be two amino acids, it can be anything. And this is energetically unfavorable, as I said before. But the hydrolysis is energetically favorable. Okay? So it will hydrolyze quite promptly, but all these molecules, so polysaccharides, nucleic acids, proteins, they all involve, if you have glucose and if you want to add glucose to glycogen, which is the storage of glucose that we have in the liver, if you want to add one molecule of glucose to glycogen, it's a condensation reaction, and then you lose water. Protein, if you want to add a new amino acid onto a polypeptide, again, it's a condensation reaction that involves ATP. And if you want to add a nucleotide onto a chain of nucleic acid, then again, it's the same reaction. It is very, very commonly such reaction, and they always require energy. But if you take a look at this chemistry, this is ATP. This is similar to what we saw before. ATP is transformed into ATP. And the delta G of this reaction provides minus 11 to minus 13 kilocalorie per mole. That's about the energy that is in ATP. It's great. It's a high amount of energy, and it's enough energy to do that. It's enough energy to do that. But it's not enough energy to do that. Because of the molecules, because the structure, it requires more energy. But still, ATP is used for that. How? You have ATP here, and the normal reaction is to cut this bond here, right? This is what you are doing here. You cut this phosphor anhydride bond here, and you liberate minus 11 to minus 30 kilocalorie per mole. But if you want more energy, you can. In very specific reaction, actually you have ATP, and this phosphor anhydride bond is hydrolyzed. And so you have pyrophosphate plus adenosine monophosphate, and then pyrophosphate is once more hydrolyzed into 2-phosphate. And the total reaction, so this hydrolyzes and this hydrolyzes, actually generates about minus 26 kilocalorie per mole. So almost the double of that, quite the double of this one. And this provides enough energy to do this reaction. So you have this polynucleotide chain, and you want to put a new nucleotide here. So first you have nucleotide monophosphate, and you must add 2-phosphate to change into triphosphate. Okay. Once you have the high energy intermediate triphosphate, then what you do is that you actually use the energy of these 2-phosphate, you break this bond, and then you can see pyrophosphate here, which is further hydrolyzed to 2-phosphate, and then this provides enough energy to actually make the new bond between the new nucleotide and the chain. But it's very interesting because ATP, it's a little bit strange, but ATP is used to add the phosphate. And then after that, when you cut this allows the reaction between this nucleotide and the chain. This nucleotide can be anything. It can be GTP, it can be CTP, it can be TTP, it can be any of the triphosphate nucleotide, but when you break this bond, it provides energy for this new nucleotide to be attached to the polynucleotide chain. But you still need ATP to synthesize the 2-phosphate. So it's a little bit kind of a very, it's a big detour to do this kind of reaction. But you need to do because you need energy to make this bond. So you can think about if you have a cell that is actively dividing, it will use a lot of energy. It will use a huge amount of energy to copy its DNA, because every time it needs to add a nucleotide, it will use a huge quantity of ATP. So this is why cancer cells, they use a lot of energy. And if you have cancer cells developing, you will really feel feeble, because if the cancer size is consequent, then they will really, really take a large amount of energy from your body just to make sure that they can synthesize their DNA. All right. And that is it for this chapter. Do you have any question about these intermediates before we go into chapter 4? Chapter 4 is a little bit more biological, less chemical. Do you have any question? About carriers? Don't hesitate. No? It's okay? Okay. Don't hesitate. So the next chapter, the chapter 4, it's about protein and structures. The shape and structure of proteins, how proteins work, how proteins are controlled, and how proteins are studied. And first we are going to look at different types of protein. Essentially, when you look at your cell, you are mainly looking at protein. I am looking at a protein. I am looking at the protein. If you look at cells under the microscope, you are looking at protein. If you look at the virus, you look at proteins. We are mostly made of protein. But there are a lot of different types. As you know, as we have seen already many times, they are enzymes. And the function of enzymes is just to catalyze chemical reactions. So you can have an example here. Living cells contain thousands of different enzymes, each of which catalyze, speed up one particular reaction. Tryptophan synthetase synthesizes the amino acid tryptophan. Pepsin degrades protein in the stomach. Ribulose biphosphate carboxylase help convert carbon dioxide into sugar in plants. So they are very useful. They are really critical for the survival of the organisms. And there are many types of enzymes. There are also proteins that are used just as your skeleton. Okay? It's the cellular skeleton, like what you have here. This is actually a structural protein. This is keratin that you have in your hair. But cells also have keratin, and it's very important for the cell to keep its shape. Okay? So the example of this structural protein is, for example, collagen and elastin in your skin, tubulin inside the cells for the microtubules and all the cytoskeleton. So these are proteins. They are not enzymes. They don't catalyze reaction. They are just structure. Okay, just like beam in your house and things like this. You have proteins that are used for transport. They are just transporters of things. So for example, you have a picture here of a red blood cells because they mention hemoglobin. Hemoglobin is only used as a transporter of oxygen. Okay, but there are many transport proteins who will transport protons and will transport phosphate, and many other type of molecules will be transported by proteins. So for example, calcium pump here, they are mentioned. So transport protein, we have motor protein. I think these are one of the most interesting type of protein because they generate movement inside the cell. So your muscles, mostly made of myosin. This is very important for your muscle to be able to contract. This myosin protein is the main component of your muscle. And you have also DNA in here. So if you have a bacteria with like eukaryotic cells and they have cilia or little flagellum, this is made of tannin. So it's all this mechanical movement. Okay? This is very important for mechanical movement. And we will see some really famous examples of these things later. There are proteins whose sole function is just to be a storage of amino acids. It's a protein, but it's used in case the cell need to get amino acid very quickly. So what you have here, you have a bottle of milk here, or a carton of milk, and you have eggs, because the ovalbumin in egg is just a storage of amino acid for the chick. And in milk, you have similar protein. This is casein. It's just a storage of protein for the baby cow or baby sheep or whatever. So they are just amino acid storage protein. You have proteins who are carrying the signal from one end of the cell to the other. So for example, insulin. Insulin, you all know insulin. It's a hormone. But insulin is a protein. A very small one, but it's a protein. Okay? And you have other proteins here. You have natrin, attracts growing nerve cell axon to specific locations in the developing spinal cord. So you have many types of protein who are signaling, who are carrying a message to the cell. Insulin is really the famous example of that. And you have proteins who are receptor. What is it, receptor? It's a protein that waits until something comes. So for example, it can be anything. It can be light, it can be another molecule, and when they are activated, when they bind their ligand, they are activated. So these are called receptor proteins. So they detect signals and transmit them to the cell's response machinery. So for example, rhodopsin in the retina, it's a receptor to light. When this protein receives light, it is activated and sends a signal through your brain. And this is why you are able to see. But you can see other receptors like the adrenergic receptor. It's actually a receptor to adrenaline. So you have many of them as well. And you have also proteins that regulate other genes. They are, for example, called transcription factors. They will bind DNA and they will regulate how gene is expressed. Okay? These are gene regulatory proteins. And you have many other proteins who have a very specific purpose. We can see here these shellfish because they mention this monellin here, which is a very specific protein that enables the shellfish to actually grip the substrate. So it's a very specific protein for a very specific purpose. So you have a lot of different types of protein, and they all rely on one thing, their structure. Structure is very important. Okay, this is going to be the next step. The shape of a protein is specified by its amino acid sequence. I will go quickly about this one. You know, you have two amino acids, you have glycine and alanine, and they form together a peptide bond, and this forms glycylalanine. Okay, and the protein, you repeat that many times, 300 types, 3,000 types. It can be very small protein, it can be very big protein. And so to remember, this is all the peptide bonds of amino acids. We have 4 amino acids, and amino acids have different side chains, and they can be very different because some side chains can be negatively charged, positively charged, uncharged, or non-polar, and this will give completely different properties to the protein. And inside the protein, there are different non-covalent bonds that will make sure that the protein keeps its shape. There will be electrostatic attractions, for example, between polar positive and polar negative amino acid. There will be hydrogen bonds. And there will be something that I haven't talked about yet, but this is van der Waals attraction. So this is attraction between non-polar molecules. And for example, here you have a lot of amino acids that have a methyl. So this is hydrophobic, so they will just stick together. And they will try to get away from the surrounding environment. So if you have here, it's a theoretical polypeptide, and you have many side chains with different properties. So usually what you have is that all the polar amino acids will usually be on the outside of the protein, because the protein is in the aqueous medium. But inside, you will have all the amino acids that have a non-polar side chain. So they will tend to gather inside, okay, and be away from the water, away from the cytoplasm. This is mostly the case in cells that are of proteins that are inside our cells. And these hydrogen bonds, they can be made in different ways. They can be made between two backbones. So this is the backbone of the chain, right? So these electrostatic attractions, these hydrogen bonds, can be made backbone to backbone, backbone to backbone, backbone to side chain, or side chain to side chain, because there are many potential non-covalent bonds that can be made between every single atom of the protein. And the protein usually takes the shape that has the lowest energy. This is very important. So imagine you take a protein, you purify it from a cell, and you add a high concentration of urea. This is urea. So you can see it will disturb all the hydrogen bond because you have this oxygen and you have the two amine here. So if you put a high concentration of urea, you will denature the protein. But when you remove the urea, the protein will very often go back to its shape. because the shape of a protein is usually the one that is the most stable, the lowest energy conformation. And why it is important, this seems very simple. Well, yeah, that's of course. But no, because they are diseases that are caused by protein being misfolded. So, this one, imagine you have a normal protein, but on some occasion, this normal protein is folded strangely, completely differently from what it should be, its normal shape. This is called misfolded prion form. Okay? It's not always prion. This case is a prion. And then what happens is that when this misfolded protein meets another protein who is correctly folded, what can happen is that this misfolded protein will trigger the misfolding of the healthy protein, which is the same in the beginning as itself. And this will amplify many times. And you'll have a lot of misfolded aggregates like this. These are called amyloid fibrils. Do you know in what disease this happens?
Alzheimer.
Alzheimer. Alzheimer disease is exactly because of that. We don't really understand the cause, but Alzheimer is you have very long fibers of these misfolded protein that accumulate in the cells. And cells die because these fibrils are very strong. They're almost like having needles in your brain. So this is a problem. What kind of other molecule? Alzheimer progressively becomes worse. So this accumulates over time. But there are other diseases which are based on the cell. You cannot really say prion protein in Alzheimer. Not really prion protein. But there is a special case of a disease in which this kind of misfolded protein is called a prion. Do you have any idea? There are a few diseases. There is one in human, there is one in cow. And it's even bad that if you eat this misfolded protein, you can actually catch the disease. So usually, a contagious agent. What is a contagious agent? What does it mean, a contagious agent? Can you give me the definition of a contagious agent? Contagious.
Misfolded protein.
No, not only. Like bacteria, they are contagious. Viruses are contagious. If you have a cold, and then if you sort of cough in front of somebody, you will get my virus, or you will get my bacteria, and you will also get the disease. Because bacteria will multiply, virus will multiply. These are contagious agents. But these prion proteins are contagious, but they don't contain any DNA. There is nothing. Okay? They have no genomic information. They are just a misfolded protein, but for some reason, they behave as a contagious agent. So what kind of disease these things happen? Do you know? It was a very big problem in the UK maybe 15 years ago, in Australia, United Kingdom, and it comes every now and then. You know the BSE, bovine spongiform encephalopathy, or Creutzfeldt-Jakob in human. So all these disease is because you get contaminated by something that trigger, by a prion protein that trigger your own protein to become misfolded. And we still don't really know how to treat these things. Okay? Alzheimer, there is no really good treatment for it. And it's all because sometimes protein can get misfolded, and misfolding is amplifying itself. So it's kind of a big problem. So, because the folding of protein is so important, then the cell has developed chaperone proteins to make sure that the protein is correctly folded. So you have two cases here, where you have one protein without the help of the chaperone, it gets incorrectly folded, but with chaperone protein, it gets folded. So sometimes these chaperones are just proteins that sort of help the correct conformation, but sometimes chaperones are just like a box in which the protein to be folded goes inside, and there is a very specific environment inside the chaperone that allows the protein to get its final conformation. So this is the role of chaperone, is to correctly fold protein. Okay. Proteins come in a wide variety of complicated shapes. So please take a look at this picture. This is the kind of space-filling model of many kinds of proteins. Some proteins are very long, like this, like a fiber, but some proteins are globular, like this one, cytochrome C, catalase. So they have very, very different shape. And this is just different. This is one protein from a bacteria, and this is, although it's very small, you can see if you actually look at how the protein is configured, you can notice different structure inside the protein. Here you can see the whole protein has a specific structure, but if you take a look, some part of the protein they are helices, and some part of the proteins they are a little bit different. So we are going to look at the very basic shape of protein. So, what is the structure of the protein? The primary structure, it is just the sequence of amino acids. This is the primary structure. The secondary structure is very small. It can be quite long, but it's arrangement of amino acids within the structure of the protein. And there are mainly two types that we are going to see here, is the alpha helix and the beta sheet. And these are very critical folding patterns. So you imagine this is a helix of amino acid, and you can see there are hydrogen bonds which are made between the backbone of each amino acid inside the helix. Okay, inside the helix. But you can see all the R. This is the back. This is the side chain of the amino acid. You can see they are all sticking outside. Inside of the alpha helix, you only have these backbone, but outside, sticking out, are all the side chain. An example of a protein that is only made of such alpha helix is the alpha keratin, the one in your hair. The other one, the other folding pattern is beta sheet, and you can see it's completely different. Beta sheet is a chain of amino acid that is associated with a nearby chain associated to a nearby chain. So it really makes like a sheet, okay? And you can see in this sheet, again, all the hydrogen bonds are made between the side chain of the nearby polypeptide chain. And all the side chain are sticking either up or down, okay? So they are on this side or on the lower side, which is also very important for how these amino acids associate with different components of the cell. And an example of a protein that is made of these beta sheets is fibroin, which is the main component of silk. Okay? This is why this is very, very strong. It's a very strong association between polypeptide chain. So this is why silk is actually quite strong. And these beta sheets, this is what happens here. These beta sheets, this is what happens here. So they are very, very, very stable configuration of amino acid. Okay. Just a little bit, just a mention on helices. You know, there is the DNA double helix, there is RNA helix also happens, you have the alpha helix. If you actually make a molecule that is composed of the same subunits many, many times, naturally this will take the shape of a helix. Helix, they are very ubiquitous in our cells, and a helix can be right-handed. You can see here, this is right-handed, and this is left-handed. Can you recognize when you see a left-handed and a right-handed helix? So if you screw in, it goes in. and if you screw out, it goes out. But left-handed is the opposite. Anyway, so this is a left-handed, you can see, and this is right-handed, so it goes like this, and this is left-handed, so it goes like this. So it's a little bit, you can look at this one here. Sort of it goes clockwise. This one goes anti-clockwise. And why are these alpha helix important? Because they are perfect to anchor a protein into the membrane. Because you know the membrane is composed of these phospholipids, so inside here is hydrophobic, okay? It's hydrocarbon, so it's highly hydrophobic. And also the backbone of this polypeptide has many, as I told you, has many hydrogen bonds. If you imagine this is all hydrophobic side chain, then the affinity for the small polypeptide for the double membrane will be very high. Just because these side chains are hydrophobic, they will associate with the membrane. So this is really great, okay? And there is another possibility in which not all the amino acids are hydrophobic, but only half of them. So the side chain of half of the amino acid is hydrophobic, and the side chain of half of them is not hydrophobic. And then what you can get is actually you get two side chains that will actually associate with each other. The hydrophobic amino acid will interact with each other, which is the green here, and the hydrophilic amino acid will actually stick out. This is, for example, alpha keratin. This is one molecule of alpha keratin. Okay. Two alpha helices associated together. And this is called a coiled coil conformation. It's a little bit difficult, you don't need to remember, but let's just say this is a coiled coil. Sometimes scientists like to name very complicated things. This is the advantage of alpha helix. Beta sheets, they can be, this is anti-parallel. It's called anti-parallel because you can see this is the beginning of the polypeptide and it goes like this, like this, like this. So each amino acid chain goes in a different direction, and this is parallel. So this is the beginning of the protein, and you can see all of these sheets, they are parallel to each other. So this is anti-parallel, this is parallel. This is just the convention. And what you have here, it's at the microscopical level. These are these things that I showed you here. These misfolded protein, these amyloid fibers. So if you repeat this one many times, then what you get is very strong amyloid fibers like that accumulate in the cell. And you can see this is made of interlocking beta sheets. So this is a very stable conformation. Beta sheets are not always bad. They are very important for the structure of normal proteins, but it's just to tell you that actually getting so many beta sheets makes the protein extremely stable and not so good for the cell. All right. Proteins have several levels of organization. As I said before, you have the primary structure. This is the amino acid sequence. The secondary structure, this is the folding pattern, either alpha helix or beta sheets. Then you get the tertiary structure, which is just the assembly of the protein as one molecule. And then you have the quaternary structure, which is many subunits inside the same protein. So one protein is made of many different subunits. And the quaternary structure is all the subunits together. But what is not mentioned is that sometimes, well, not sometimes, all the time, is that proteins, you imagine this protein here, this is a bacterial protein called the catabolite activator protein. You can see this protein has beta sheets, alpha helices, but it also has two domains. There is one domain here, and there is one domain here. This domain binds cyclic AMP, and this domain binds DNA. And it's very important because this domain communicates to this one. When this domain binds CAMP, cyclic AMP, it will change its shape and it will promote this domain to bind DNA. So the two domains sort of communicate with each other. But this domain association is not included into the primary, secondary, tertiary, catenary structure. It's just another level of organization. So proteins are made of sometimes many of these domains. And they have very specific purpose. For example, this one binds DNA, this one binds cyclic AMP. And this is different proteins. Some proteins are exclusively made of alpha helices. Some proteins are a mix between beta sheets and alpha helix. And some proteins are exclusively made of beta sheets. When you have something like this, it's called a beta barrel because it looks like, you know, something you put wine inside. And inside is hollow. Okay. But you can see here that you have these alpha helices, but you also have these links between these links here. These are called unstructured regions. And they are as important as polypeptides that have a specific structure. These unstructured sections of proteins, they are very important for binding because they can cradle other proteins like that. But they can also be used to actually link two different domains together. So you have this green domain and the blue domain, and you have this unstructured region that is very flexible, and it is used as a tether to make sure that these two domains can interact together, but not always. And this unstructured region can also be used to actually bring together different parts of a scaffold protein. A scaffold protein is a very big protein that binds many different things, and then when it does so, all these different things that are bound to the protein can actually be gathered together and react together. So, unstructured regions are very important, but the problem is that maybe I will not mention that. Okay, so let's imagine a protein made of 300 amino acids. 300 amino acids. How many possibilities are there? How many different proteins of 300 amino acids? 300 amino acids is not specific, it's not really big. Okay? It's an average size for a protein. So if you imagine a protein made of 300 amino acids, how many possibilities are there? How many proteins can be made with 300 amino acids?
20 to the power of 300.
Yes. 20 to the power of 300. The amount is really huge. Okay? 20 to the power of 300. This is really huge. And they are not that amount of protein, of different types of protein. This is too large. And yet it's only for 300 amino acids. Some proteins are larger, 400, 500, even 1000 amino acids. So why do you think there are not so many proteins? If you look at many different types of protein, you will not find 20 to the power of 300. Why not? This is what it is. Few of the many possible polypeptide chains will be useful. But why? You know, of course, that it's not random, right? It's not random. Cells do not make protein at random. Well, sometimes they do, but most of the case they don't make protein at random. But why? Why among all these 20 to the power of 300 possibilities, why only a few sequences will be used?
By natural selection.
Yes, by natural selection. But what does it mean?
an individual, a cell which produces not useful protein will be die.
Yes, exactly. So during evolution, proteins have appeared, proteins have been made by cells, and some of these proteins, they have a good function. It was useful for the cells. So these proteins were kept and they evolved. So if you look at a bacteria and if you look at humans, some proteins are actually very similar because they had a function and they were selected. But when proteins do not function well, they are completely random, then they have no function or they have very bad function. So these are selected out. So what we have in our cells is all these proteins that have been selected during evolution to have a function. And function depends on the structure of protein. So this is why the amino acid sequence is relatively fixed. Okay? So this is why it's certainly not random. And because of that, you can actually see several proteins families. If you take a look at two serine proteases, so this is a protein that will cut other protein. Okay? It's a protein that will cut other protein. For example, elastase. It's a protein that cuts elastin. And you have another protein that has a different substrate. And the substrate is a little bit different, but not so different. In both cases, both elastase and chymotrypsin, they will cut where you have a serine in other proteins. So this is why they are called serine proteases. And even though they are different, okay, one is in bacteria, the other one is in Drosophila, you can see that actually the structure is quite similar. And the domain, the kind of sequences are relatively well conserved. And the domain that is accepting the serine to be cut is actually very similar. Okay. So there are different families who have a very, very similar structure. And you can check the same protein in humans and the same protein of a same family in a different cell, even in bacteria, you will get very similar structure. Not exactly the same, but very similar structure. And some very large protein, like this CAP protein, you remember I told you this one before, this CAP protein here. This protein that can bind DNA, when it binds to cyclic AMP, actually it does not act as one monomer. It actually acts as two dimers of the same unit. And you can see that the protein is really two subunits of the same CAP dimer. And you have another protein, whatever the name, it's actually a repeat of 4 subunits. And you have sometimes even many more subunits to make the protein. So this is also very important for the function of the protein. This is hemoglobin, by the way. So you can see hemoglobin is actually a tetramer, 2 alpha globin and 2 beta globin. And the whole hemoglobin is a tetramer. What is this one here? Can you guess what is this? You can see, so all this is amino acid, right? All this you see alpha helices there. You don't see much beta sheets here, but mainly alpha helix. What is this? It's
Iron.
It's it looks very big for iron. It looks very, very big for one little. So what it is? Yeah, you are right. Actually, inside here somewhere, it's probably not drawn, but you have iron. But what is there? We will see later. Okay. Proteins can assemble into filaments, sheets, or spheres, which is really great because proteins, they really self-assemble. If you imagine one little subunit and has one single binding site, so you put 2 of these subunits into water, then you will have a dimer. They will recognize each other and they will bind. However, if you imagine another protein with two binding sites, where this can bind to this, and if you add a lot of these monomers, then what you get is that they will form a helix. But if you have binding site at a slightly different conformation, then they will form a ring. So proteins, they self-assemble depending on their structure. Okay, so this is the beauty of the whole protein thing. And you can see here, this is tetramer. It's a trimer, 1, 2, 3, of a protein that forms one of these, this filament here. It's a filament of keratin. Okay. So one subunit, depending on the protein, can form filament, for example, keratin, spherical cell, they can form a sphere, or they can form a hollow tube. One example of hollow tube is, for example, microtubule, which is a different protein that is the cytoskeleton of your cells. And spherical cells, do you know any example of that? Of a protein that is made of different subunits that associate into a ball. For example, and I think it's very beautiful, I think I already showed this picture in the previous chapter. This is this one here. So you can see many different subunits, they are all linked to each other. This is actually a virus. And inside you have the genome of the virus. It can be RNA or it can be DNA. But you can see a very almost perfectly round shape made of different subunits of the protein. And the great stuff is that when these proteins are expressed in your cells, which is not great, it's bad for you, but when these proteins are made into the cell, they self-assemble, okay? Because of their structure, they will recognize each other and self-assemble into such an organized structure. Okay. Most of the proteins we have seen so far, they are, the subunit like this is globular. You see, it's like a round. This one as well is like a round. This one as well, it's making some sort of long chain, but one protein itself is globular. It's just like a little ball. But some proteins are completely different. And this is, for example, what you have in your skin. This is a molecule of collagen, and you have a triple helix of collagen. This is one molecule of collagen. Very, very long, 300 nanometer, but very thin, 1.5 nanometer. And it's very, very long for its size, and it's actually a triple helix. This protein is not globular. It just looks like a string, okay? And you have another type of protein, which is different, but one protein, so one chain of polypeptide, is also completely like a fiber. It's a fibrous protein, and this is elastin. And you can see these elastin, so this is very unstructured polypeptide. It's not beta sheets, it's not alpha helix, it's just completely unstructured region, but they are linked to each other. So this gives the property that it can be stretched, and this is very important for your skin to be elastic. And once you relax, then it goes back to its own structure. So this is another example that even though the protein may not be highly organized, it's also very important for a specific function. Okay. So we have seen that what is responsible for the structure of protein is mainly all the non-covalent interactions that every atom does with the structure of the protein. But there are some chemical modifications that make sure that the protein will keep its structure once it is made. For example, this case is you have two amino acids here that have a sulfur-containing side chain. And when you have two sulfur-containing side chain here, so you have the protein here that has been folded according to its non-covalent pattern, if you want. So this is a cysteine here, you have another cysteine here, another cysteine here. And then what you can do, you can actually oxidize these sulfur to actually make interchain disulfide bond and intrachain disulfide bond. And these actually are very important because they will make sure that the protein will keep its shape. Can you guess what will be the purpose of this kind of molecule? A molecule that contains a lot of disulfide bond. What may be the purpose of this kind of molecule, of this kind of proteins? Huh?
Hair.
Hair. No. But there is an idea. So hair, it is outside, right? It is outside of the cell. Okay. But hair contain quite a lot of sulfur amino acid, but not so many disulfide bonds. Not so many disulfide bonds. What kind of circumstance do you think that sometime a protein with disulfide bond will be useful? Keep in mind that disulfide bond will stabilize the structure and will make the protein very resistant. For what purpose this could be?
Enzyme.
Enzyme, yes, enzyme. But for what kind of enzyme? Why would you want to make sure that your enzyme has a very stable and strong structure?
To attach to very specific molecule.
Yes, although this is really not really attachment. This will stabilize the protein, but a protein without disulfide bond could very well nicely attach to other protein. The attachment may be even more stable with SH, yes, with disulfide bond, but in one case this will be useful. Think about a cell, making all sorts of molecules going to different areas in the cell. And there are maybe some cases where, I really have to make a protein that is very strong there because something may happen to this protein and it may be very sensitive to being degraded. So, when could this happen?
In lysosome or peroxisome.
Yes, for example, in an environment that is a little bit aggressive, into the cytoplasm, there's no problem. It's relatively normal environment. But what if you actually if you make a protein, if you make an enzyme that you release outside, for example? Some cells are secreting enzymes. So, for example, you have enzymes in your saliva that have an effect and that are killing bacteria. Not very, very efficient, but these kind of enzymes that are secreted outside the cells usually contain a lot of disulfide bond because they will be outside of the cell. And outside of the cell, it's more aggressive than inside, so it is useful for these proteins going outside to have a very stable structure stabilized by disulfide bond. And the same is also true for some parts inside the cells that are very aggressive, like in lysosome or peroxisome. Okay. And I will stop here for today. Thank you very much.